Hi, I'm Professor Marloes Peters, and in this short video I'm going to talk about what the future perspective of molecularly imprinted polymers is. So to recap, if you haven't watched any of these videos on molecularly imprinted polymers, or MIPs in short, these are porous materials that are polymeric materials that contain binding sites with very high specificity and selectivity for the target they are designed for. So there are instances now where these MIPs can really compete with antibodies in terms of this selectivity and specificity. And in that case, they would offer the advantages of much lower costs, even though that's the question whether that's the case at really larger scale, from ethical reasons. So, for instance, there's still a lot of animals being used to produce antibodies, and also they have enhanced robustness, so they can operate in conditions that normal natural receptors can't. So think of elevated temperatures or low or high pH, etc. So many advantages there, including the versatility. So for instance, we can use them from something as small as ions up until larger macromolecules. But why, even if we can achieve, the similar affinity as a natural receptor, why are they not being used commercially? So this is what I'll talk about in this short video, and I'll go into detail into 3 different topics. The first one is the sustainability. So think if you were a business and you would have to sell this, traditionally these MIPs are marketed as plastic antibodies. So the word plastic has a lot of negative connotations with it, because people think of landfill, they think of plastic bags that end up contaminating the environment, and they don't necessarily think so much about the prospect of that there's a lot of polymers that are being used for healthcare applications. So think, for instance, if you would wear contact lenses. So they are used regularly, and they can also be produced from sustainable sources. So obviously it would be very important to look at what they are made of, these polymers. So the monomers, so that's where you make the polymers of, they need to be sustainable. So they shouldn't be derived from oils. There are a lot of people looking at, for instance, other materials, such as, for instance, things that could be waste products or things that might be biodegradable in the longer term. So the sustainability, we could argue then, if that's not necessarily a problem, they would be able to break down. So bear in mind, these are cross-linked materials, so that could potentially be a problem for breaking this down. The sustainability aspect also comes to what I said, this ethical concerns that you might have. So if you think of sustainable production, because of these molecularly imprinted polymers being completely animal-free technology, this also enhances the sustainability aspect because you're not using biological resources. And also because you're not using biological resources. And hopefully that should also add to increased reproducibility, because that can be another problem of biological materials, which also, if you look at healthcare applications, might be able to trigger some kind of response. So if you look at net zero targets, in principle, these MIPs should be an appealing alternative. The second point relates to the functionality. Most of the time just see this as antibody mimics. So they would replace, if you have a traditional immunoassay where you use these natural receptors as recognition elements, you would simply replace those with these molecularly imprinted polymers. And I think that's actually doing the materials a disservice. Because you have polymer materials, they're much more versatile. It's much easier to modify them. You can include lots of, for instance, probes to check interactions. But also you can have dual functionality. And that's where definitely this field is emerging towards, where they're looking at what else can we do besides simply the sensing. And for instance, we exploited this dual functionality to not just sense a certain receptor, but to also achieve drug delivery and make sure these materials can really enter the nucleus. But for instance, you could also modify the surfaces of other materials. So you could use them for cancer treatment, you could use them for imaging. So there's far more that goes beyond this traditional antibody replacement. So even if you would use them as antibody replacement, you might be able to add additional things in there. So I mentioned these fluorescent probe, which makes it really easy to detect, but also a redox probe, etc. It's very hard to compete with antibodies or enzymes, which are traditionally very much established. So in order to really make sure that we enter a different part, so a niche in the market, we should look at other functionalities and where MIPs can have applications that these traditional materials can't. The final and the third point relates to the biocompatibility. So in the previous point, I went into we can have this dual functionality, so we can have additional functionality of MIPs. However, the thing that hasn't been studied properly, obviously if you're using this for in vitro diagnostics, this is not a concern, but people are starting to consider this for clinical applications. So there's no clinical trials out there, and even if you look at the animal data, this is quite limited. So there are some reports, but there's also some conflicting results there. And generally, people tend to say that this material is highly biocompatible. But what they often just do, they simply just mix it in with a cell line and they check the cytotoxicity afterwards. So that's not sufficient if you want to look at other aspects, if you want to look at if they potentially would invoke an immune response, the clearance rate, how it's cleared from the body, the toxicity. And for all of this will be highly dependent on the material. So you can't just assume that you test one particular molecularly imprinted polymer. If you want it for biocompatibility, then it needs to be like a nanoparticle system. But it will highly depend on the size of that material, how it's cross-linked, what monomers you use, what ratios, even things like the initiators start to play a role. So there's no standardized protocol into place to actually check this biocompatibility. And even though it's claimed often in literature, we really don't know how it behaves that well. And especially when we look at like a longer term, because we are working with a cross-linked system. So the issue that you might get is that you get some degradation there and that you do have some toxic products that do leach into the tissue. So here I went into brief detail into these 3 different points. We are working on a future perspective on this particular topic. And do keep an eye out when that comes out, where we'll go into more detail and what kind of solutions there are to overcome these 3 different problems. In general, I would say we need to focus much more on some computational modeling, so rational design of our system so we understand the system a lot better. And we need to have a look at how we automate everything, so for both from the design to the production and screening for the affinity. And finally, the biocompatibility needs to be thoroughly studied in order to check whether these MIPs can actually have interesting healthcare applications, for instance, for treatment of certain diseases and as therapeutic. Thanks for watching, and do have a look at our playlist, Bio-Inspired Materials, if you have an interest in this particular topic.